series is sponsored by an endowment generously given to the department by Tom and Nina Lee. Tom Lee was an entomologist in the department who began his career here in 1958. He was in the department for more than 30 years and very generously sponsored this seminar series, which is to recognize and honor the achievements of distinguished alumni from the department, the graduate program. We've had a number of speakers over the year who have given us lots of great information, and tonight's speaker is Dr. Brian Fisher, and he'll be introduced by Phil Ward. Thanks for coming.
That's right, here you go. Thank you. It's my pleasure to introduce Brian Fisher. The first thing you need to know about Brian is an ironic fact that he hails from Normal, Illinois. And that's the last word you'd think of when you consider Brian. You know, he's just this extraordinary fellow, a great adventurer, an innovator. He's brought tremendous creativity to the study of ant biology and taxonomy. And he's been through more adventures than the rest of us put together probably in his lifetime. He's done fieldwork in all corners of the globe, from Central and South America to Africa to Madagascar, which we'll talk about tonight, to Asia, China, Australia, and very often under very trying circumstances, literally near-death experiences that he's had in some of these remote field locations. At the same time, he somehow manages to always extricate himself. Like the legendary cat, he's gone through multiple lives, and yet he's still with us. Brian has also been tremendously innovative in other aspects of ant biology. So when he's not away in the field, he's been organizing a tremendous website called AntWeb, which has basically revolutionized the way we disseminate information about ant diversity. He organized something called the Ant Course, which is one of the most incredibly successful and popular courses on ant biology. It has people at the door begging to be admitted to this very popular course that Brian essentially single-handedly instigated and set up. I'm supposed to give a potted history. Besides coming from Normal, Illinois and being far from normal, he did his undergraduate degree at the University of Iowa, had a master's degree at University of Utah, a brief stint at the Field Museum, and then it was my good fortune to attract him to Davis in 1993 or thereabouts. And when he came to Davis, we infected him with a virus, which transformed him from an ecologist to a taxonomist. One of my proud achievements. And since that time, he's been focusing his efforts on biodiversity, the taxonomy and systematics of ants of the world, whereas previously he'd worked largely in the area of ant ecology, worked on ant-plant relationships in South America. I don't want to take more of your time. We're here to hear Brian, so I'll just simply say he'll be talking tonight about his adventures in one small portion of the globe, but an area that's just an amazing part of the world that's full of life and problems, and the two seem to go together, and that's the island of Madagascar, an astonishingly rich area, biologically diverse, high levels of endemism, enormous environmental problems, and Brian today will share some of his adventures on that island.
If you all can hear me, I'll just speak next to the microphone. Thank you, Phil. The virus is still active. I've been treating it unsuccessfully, so tonight I'm going to go deep into the world of taxonomy a bit to try to infect you by the evening, so be careful. If you're not wanting to be infected, please move to the back. So the world of ants is important to me, and these, if you're an ant lover, you look here and go, oh, how beautiful. Look at these beautiful mandibles. They all have beautiful lifestyles. They actually are what we call the glue that holds the forest together. And when we try to convince the public about ants, we remind the public that it was the ants that invented farming. 50 million years ago, ants began a relationship with fungus. They bring plant matter. Our neighborhood cypromyrmex up around Lake Berryessa does not gather leaves, but gathers up caterpillar frass to feed their fungus. You can go check that out some evening, just climb up on the hills around Cold Canyon, and you can find our local fungus growing ant. So ants invented farming. We probably have a lot to learn about farming from this relationship of basically growing a monoculture for the last 15 million years. Ants also invented the refrigerator, and that gets really excited the fraternities about, beers and draft beer service here in the colony. So basically what they do is they take these young, enclosed workers and stuff their social stomach. See, ants have two stomachs. They stuff their social stomach with honeydew until they bloat out, and this is actually the stomach here, basically, just swollen up the crop. And the ants hang from the ceiling, and during those times of hunger, other ants come along and are fed from these ants. And one of the most amazing ants to describe to the public are the army ants. They're like a leaderless Wolverine pack that sweeps through the forest, eating everything that can be found. Now, because of that, they often have to change their nesting sites. And to do that, they have to actually build a nest each night. And they do that out of their own bodies. Like a scarf that's woven together, they hook arm to leg, and weave their own nest each night. Now I thought, wouldn't it be great to be able to take this amazing problem-solving ant to San Francisco and have on display? So I took my vacuum cleaner from Home Depot and went down and sucked up a colony and had them on display for about 12 months in San Francisco. It's an interesting story how to get them here. Collecting was the easy part. Getting them here was the hard part. And actually feeding them. They ate 25,000 crickets a day. But the kids loved it. They loved to see all those 25,000 times 6 legs being ripped off and carried into the nest. But also they exhibited amazing solving problem that ants can do. Ants working in small groups, they can actually solve amazing problems. And that kind of swarm intelligence is actually useful in many parts of society. To help explain why ants are important, it's good to understand their role in the environment. But to me, I feel we have a long way to go in explaining the public. To me, what I really want to see is what we have in the bird world. To me, the competition are the birds. We don't have any apps, really, on identifying the local ants. We don't have a field guide of ants for every single country in the world. We don't have companies that can take you on an ant tour to Madagascar or around. So to me, this is the goal, is to find a way to get enough information together and enough interest to create this kind of industry of excitement around the world of ants. And in a scientific way, instead of seeing yet another paper in Science or Nature about global diversity of a vertebrate, we want to start seeing global diversity studies on the distribution of species of ants. We want to have Android apps to be able to walk around and say, hey, what are the ants nearby here? Download on your iPhone a picture guide of the ants. So success for the taxonomists and ecologists will be when one day we have the public, just screaming to have these resources. And they turn to these pictures and go, oh my gosh, this is the best ant porn. Check this out. These ants are interesting, and each one has a different story. You want the people to connect to that story. But it's not really maybe in the cards for the next few years, but we have hope, I think. But we have problems. Now, there has been this demise of field-based biology, and this is great in the sense that who this lecture is in honor of, in a sense, was such a broad-based entomologist who supported the study at the organismal level. But we've seen that in most cases, we're moving to the discovery-based science to be at the sub-organismal level. And that's actually impacting who knows about ants and to be able to identify ants. If you look at recent contributions to GenBank, for example, which is one of the largest biodiversity databases, you'll see that there is a surge in new information coming in about life, but most of them is unnamed. In fact, the bulk of taxa being submitted to GenBank are unnamed, which is actually just what we have for bacteria. So most of life is moving to the same platform in which we understand bacteria, which means that most of biodiversity that we're learning about right now is simply GPS coordinates tagged to DNA sequences. So we can refer to this as dark taxa. So this dark taxa is where we're moving in terms of our kind of research on life on this planet. But the question to ask ourselves is, how much biology can we do without actually knowing the names of those taxa? So who's putting names on these taxa? Who's putting names and actually delving into the discovery of new species? And that, of course, are taxonomists. Who's a taxonomist in the audience? We have a few. And that's great, yeah. By the end, I have more. Now, these taxonomists are asking very simple questions, right? They're asking what living things exist, where do they live, how are they related? And for me, that will be going to this region, the Southwest Indian Ocean, where you have the most amazing biogeographic kind of experiment at your disposal to study with all the different ages of islands and regions. I go there with teams, we chop into rotten logs, we dig in the ground, we put out traps. And taxonomists come back with their holy grail, their specimens. Now, traditionally, taxonomists would go out in their youth, and they'd come back in their office, and then they'd shut the door. And for the next 30 years, they would describe a few species. They would measure ant heads. They would not talk to their colleagues. They would not worry about conservation. What their focus was on was producing this book, this monograph they call a revision of a certain group. Now, they didn't care anybody read this because they felt their legacy really was that they were actually leaving behind type specimens. These are the types that bear those new names. Now, that legacy I found to actually be very fragile. After visiting many museums in Europe, I found that most of these collections are completely forgotten. Even their cabinets are turned against the wall. For the last 250 years, since the time of Linnaeus, I estimate about 90% of all these monographs, these taxonomic works, these products of those 30 years being shut inside an office are never used. They're never used, therefore they're never valued, they're never appreciated, they're never incorporated into any field guide at all. And that means nobody has any idea about these things. Nobody has any idea even about the ants maybe in their backyard. And they go to museums where taxonomists used to work, and they think of them as simply as a warehouse of dead creatures. They don't look at museums as actually an access point for understanding the world around us, an access point for relating our life into our needs as society. So maybe we should just give up and consider that taxonomy is an old field and is dying. Any dying taxonomists here? We have two candidates. Well, I want to convince you that taxonomy is not dying, that we're in this incredible breakthrough. But, as Phil mentioned, taxonomists are dying. Not me, personally, but we have a couple other ones. But the point is that we are losing taxonomists. For the project in Madagascar, where we ship specimens to 180 collaborating taxonomists, we still have half the insect groups without a living taxonomist to look them over. Well, does this matter, you say? Well, if you study these, that's the evil bird that we're competing against, less so. Since the time of Linnaeus, most bird species have already been described. So, not a problem. But if you like these things, that's an ant, you're in trouble. The bulk of life are the arthropods. And we have maybe discovered maybe 15% of all the arthropods on the planet. That is, we barely have touched the surface. How is it that we have kids that are more excited about discovering life on Mars than right here in our own backyard? How is it that recently California came up with $1 billion to fund a telescope to look at a rock they'll never visit, that will be here in 50, 100, 1,000 years from now. Meanwhile, every day we're losing species here on Earth. In 50 years, how many species will have been lost why we don't support the discovery of life on this planet? So again, you're asking, why does this matter? The bulk of life are these small things, are the arthropods. So let's review. So we have lots of undescribed species. We have fewer taxonomists. Phil's dying. And on top of that, we have this suicidal experiment. We have massive land transformation and climate change. And this, I think, for the first time, the 7 billion people on Earth may realize that there is a connection between what we do and the environment. We have realized that maybe climate change is real. This is some of the most convincing evidence I've found. So the people do realize that they have to start understanding our impact on the environment. But do we have a tool for that? Is there a way to do that? Is there any way we can really make a decision about what that mine will do and how they could offset that impact? We really don't. What we really need to come up with is some type of like Dow Jones index to the environment to really have a holistic sense about our impact on the environment. Related to that is bioliteracy. Bioliteracy. What is bioliteracy? Now, if you're illiterate and you see a stack of books, what do you see? At most, you'll see thinly sliced stacks of firewood. You won't understand the value of that museum, of that library, to the future of society. You won't realize the importance. Now, if you're bio-illiterate and you see a forest, it's just a green blob to be cut down, to be cattle on. You'll have no idea why we need that green blob if you're bio-literate. So we need new glasses. We need to find a way at the same time to increase literacy. We need to find a way to increase bioliteracy. And that means we need to increase the number of taxonomists. We need taxonomists that are different. We need taxonomists that are working differently. They're actually becoming advocates. They're actually disseminating information about what they discovered. And this was the goal for the project that we started in Madagascar, actually started during my PhD here. I thought it was a small project. But to actually, in a place like Madagascar, can we actually rationalize and actually demonstrate in a short time phrase the importance of understanding even ants, even other insects in such a poor country like Madagascar? Now, Madagascar is a special place in the world of conservation because of its geological history and geographic placement. It broke away from Africa, along with India, about 120 million years ago. India broke away 80 million years ago, leaving Seychelles, hitting Asia, forming the Himalayas. The long isolation has resulted in high endemism. All 106 species of primates are endemic to the island, the lemurs. By chance dispersal events, animals have arrived, like the vertebrates. Is it a cat or a dog? It's a fusa. In camp, we had these chickens disappearing. I was like, who's taking our chickens? And I tracked it down, and this is the chicken foot disappearing in the fusa, and that's who was eating our chickens. This was the closest I've ever gotten to a fusa. And 500 species of amphibians and reptiles are endemic, like this Europlatus. But more striking, if you were in Kenya and jumped over to Madagascar, you'd be shocked at what you don't find. There are no venomous snakes, no ungulates, no elephants, no giraffes, no pythons, but there are boas. And no dominant ants of Africa, like the driver ants, Dorlus, and the weaver ants, Ecophylla. This is a picture of actually the largest photographed Central African python. We had mutiny and we didn't have food for 6 days. So we had to kill it and eat it, sadly. But if you like ribs, let me tell you, we had ribs for about 3 days. But it was sad to eat the largest known Central African plant. And even the ants are spectacular. Rare lineages elsewhere are abundant and diverse in Madagascar. In fact, it's not just the interesting number of species you find, it's actually the interesting lineages you find in Madagascar. So to understand ants as a whole, you best include an understanding of Madagascar. Geographically, you'll find on the east coast, in the east escarpment, rich rainforest, and in the west, dry forest, in the southwest, a most amazing desert environment with these kind of Dr. Seuss plants. The people arrived quite recently, about 2,000 years ago. They arrived, we think, from Indonesia. They arrived with some of the most destructive land use practices in the great tradition of recent colonization events. They brought they brought rice cultivation from Asia and cattle herding from Africa. The end result is massive deforestation. This kind of famed gallery of baobabs is actually a famed photo of deforestation. The baobabs are the only tree left in most of the areas in the west. It's the only tree they don't cut down when they deforest. Less than 10% of the forest, the natural habitat, now remains. The white areas are basically exotic grasses that are burned annually. And these are the little patches that remain. The government was positive about making change. They asked scientists to come up with a plan based on diversity to actually choose which of the remaining patches could be put together into a network of conservation areas. They turned to all the scientists working there and who had data ready about inventories about these different locations. They had bird data, of course. But as it turns out, birds are the most inappropriate group to use. Only 70 species of forest birds are basically found throughout the entire east of Madagascar. There's actually no data there, no information to choose between these various species, various patches of forest. While the insects and the arthropods, from millipedes, scorpions, and to ants, see the world on a much finer scale and capture different parts of the story of evolutionary history of Madagascar and provide a much larger data pool for making these choices. But traditionally, these groups are not included. Why? They're thought they're too numerous, we don't have methods, taxonomists are dying, and nobody will care, basically, if we exclude them. So I set off to do an arthropod inventory to try to capture representative of the general arthropods community of Madagascar. I designed my strategy of collection based on the intercept of bioclimate, which is water availability, lots of rain here, 12-month dry season here, and the intersect of soil, geology. So chose about 250 locations across the island to inventory in a standardized way to create a data set to make these comparisons. Well, that's easy to do using a computer screen. But going to Madagascar is a different story to get to those patches. Logistics, you never know until you're there how to get there. You never know how to actually even find information about the forest until you get there. So we've become experts in a sense of vehicle recovery. Vehicle recovery involves how you get out of your picnic site to continue on. How you actually we always work in the rainy season as to how to keep moving through the mud to get to these forest sites. Some of it involves traversing lots of rivers in various ways. We first tried this balloon technique where we blow up a balloon and drive the car on it. You can blow it up using the car engine. It's quite fast. But if you've ever been in a swimming pool on a little raft and had the little raft go, pring, that's exactly what happened. So we switched techniques. So same principle. We carry these little lightweight drums along. It looks like that on the car. And we swim a rope across, and we just pull the car across, and it works. That day alone, we traversed 10 brackish water systems to go through these kind of no-man land, which is the burned grasses of Madagascar. And we've also included lots of other techniques of travel. I just wanted to say I just went to this incredible site called the Mackay, and it's the first time I actually had a positive experience working with a film company, because they brought transport. They brought a helicopter and balloons, and we could go anywhere. It was fantastic. The film will be coming out in a few months, so it just premiered in Paris, and it'll get to the States. It's called Mackay. Beautiful. But we don't usually always have a helicopter, and this is just last year's trip. I thought I'd show a couple of pictures. Once you drive somewhere, the fun begins. We spend more time getting to a location than we do actually in the locations. Because if you can drive there, people can get there. Basically, you're going to places people can't easily get to. You can almost put a probability on every single tree in Madagascar about how long it will live based on its accessibility and whether or not the soil can be farmed. But here we began after driving on a big river in a boat. Then we hiked 50 kilometers through various muddy river systems and ended up kind of in a frontier community and spent the entire day looking for dugout canoes, and then went upriver. Of course, if we can go upriver, they can go upriver. So we had to go up to parts of the river they don't want to go up to. So we worked our way up 12 of these large river waterfalls by pushing our canoes and carrying our canoes and our chickens. We always have chickens. And we landed in one of the last lowland rainforests in Madagascar, where all the Indri, the largest primate there, is not afraid of humans. And we set up camp, and we begin our kind of theater event of research of our methods. We'll do leaf litter sifting. This is actually a technique kind of developed during my PhD. And then we do beating, pitfalls, malaise traps, and about 3 other kind of trap methods, including yellow pan, for example. And just running around looking for ants, which is fun. And we have sometimes colleagues like Christian Peters working on behavior and reproductive biology of the ants we find. And here's Christian Peters watching his catch of live colonies. He just brought another batch of these to Paris on my short transit in Paris yesterday. So Paris has its advantage that they don't control live ants coming to the country, unlike California. I also worked with a team to get up into the canopy, which is very inaccessible. The Radeau de Cime, it's really a group of adventure sports people who love balloons. We blow up the balloon at 3 in the morning. I jump in this little luge, and you'd radio up and say, hey, take me to that tree, and they drag you through the tree trying to collect ants. It didn't really work. But it was fun. But this was more effective. This is like carnival balloons with pantyhose, and they drop it down on the canopy, and they drop you there, and you live in these tents. We stayed there for a week, and we set up traps, and actually were right in the canopy. And that was very effective for actually inventorying at least that patch of the canopy. I found it much easier than climbing, but Bonnie here, she's going to climb. But as to no surprise, insects are easy to collect. You get a lot of them. In fact, we were collecting about 1 million specimens a year. We had to find a way, if we were going to compete with birds, compete with the reptiles, we're going to have to find a way to process them to get information into the hands of conservationists and into the hands of taxonomists. So we trained a large team of about 15 kind of parataxonomists that specialized in different groups. And that came like a nucleus around which we created an interest in entomology in the country. And we run courses and training facilities to pick some of the best students before they get into graduate school or undergraduate school, and to move them into entomology, encourage that development in the country. So that kind of training began to grow entomology. So I developed a national museum of entomology in the country. And this was thanks to the Ministry of Education, which donated the land. And we just actually put solar arrays on it this year, and now it's actually off-grid and the largest solar array in the country of Madagascar. Can't see all the solar panels, but they're around. So this is located in Parks and Bazaars. If you want to do research on insects in Madagascar, you can actually work with us. We have 2 floors of classrooms where we teach. All visiting scientists teach courses too. And we have 2 floors of research and residence up here. So we've been collecting, shipping specimens to about 180 taxonomists. And we're saying, let's have some results. So we contact the taxonomists in Poland, Australia, and they say, well, your specimens are right here on the table. And we haven't finished yet. How many more years do you need? Oh, 10, 15. We'll have some results, but there's a problem. How can you justify all this work, all this collecting, if you have nothing to return back to the country? And on top of it, time is running out. With over 90% of the forest gone, using analysis of species turnover of those insects we've collected, with all the tumulent at UC Berkeley, we predicted that 40% of the species may already be extinct in Madagascar. And deforestation is not declining, it's increasing in Madagascar. So time is running out. And we've only maybe stopped finding new species is when all the forest is gone. So how do we change how we do taxonomy to make it more effective in the sense of impacting more people in terms of results? And how can we speed it up? So how do we change the simple model of being alone in your office for 30 years, measuring and then producing a monograph just before you die? Well, we have to make more people involved. We have to actually democratize science. We have to democratize access to the necessary information to do taxonomy. And that is access to type specimens and access to the literature, which are hidden. Nobody knows where those types are. They're in Europe. Who can afford to go to Europe? Where's the libraries that have these kind of 4,000 different journals that have published taxonomic works, even on ants? Well, we set off trying to create a digital infrastructure online for all that, and that is what AntWeb is. Our goal is to have a picture of every ant. Our goal is to have access to the ant catalog, linking to all the original literature. So you can actually now go there, see an image of the type specimens, and have access to everything to get you in the door of doing taxonomy. So now it's not just those at those select museums. Anybody can now begin, even in Madagascar, participating as a team in this taxonomic work. Now, creating AntWeb had a very focus about democratizing taxonomy, but actually 99.9% of its users aren't taxonomists. They're actually the general public. In fact, the 3 countries with the greatest user group is India, Brazil, and the United States. And what they're actually using is our Google Maps to find species that are in their countries. So we actually teamed up with Google Earth about 6 years ago to change Google Earth so that data like ants could be displayed on Google Earth. And it was actually a really long story about how to get their attention. It actually required describing an ant after Google, having that ant described and pictured in Newsweek, and then getting a call from Google. An ant taxonomist could not get in the door at Google. But talking to Google via Newsweek magazine worked. So they changed Google Earth. In fact, if you've ever gone to a picture or whatever on Google Earth and had it balloon out into balloons, that's our contribution. And that's how biodiversity data is displayed. And you can actually visualize by place your backyard. And this allows more people to access information based on where they are interested. Their backyard, where they've been, what they've seen. They don't have to type in a name. They can just view a place. Another aspect of democratizing taxonomy is changing the language. The language of morphology takes years and years to understand. But there are other characters, including like DNA, which every graduate student knows, easily learned, And that means more people can contribute to a growing knowledge about ants by contributing DNA. One tiny aspect of that is the DNA barcoding project, which allows easy access to massive sequencing. And that's what we've done. This is an older slide when I sequenced 27,000 specimens. But in fact, I just brought back from Madagascar 3,000 more plates of ants to be sequenced. But basically, it's cheap enough now we can sequence every collection event of ants in Madagascar. And we can use that in amazing ways, especially in the world of ants. First, it's given us access to males. Males fly. They have wings. This is a male ant. They are captured often away from the workers in malaise traps, and we taxonomists have just basically forgotten about them. For example, there's no key to the genera of ants of North America based on males, for example. There's not even a key. So males are not part of the story in our comparative biology of ants. But they are now, they can be now, because we are easily linking through genetic barcoding the males to species, understanding their genetic limits, and soon we'll have a key to all the genera of Madagascar based on males. We're also having other interesting discoveries of where taxonomists, based on morphology, because of convergence or differences of castes, have made mistakes. These three species of Mystrium, for example, I never doubted there were three different species, actually. But our DNA results came back and said, in fact, our first run showed that these had the same sequence. And I said, well, I guess DNA is not going to work all the time. Ran it again, and sure enough, these had the same sequence. And so we went back to Madagascar and dug up. These were subterranean ants, and dug them up. And whoops, here's Christian Peters, who was with us. And here they are, living together, those two species. But they're not, actually. These are the workers, and these are the queens. This group of ants, the Mystrium, has this phenomenal kind of almost new reproductive caste that's launched a whole series of studies on the evolution of winglessness in ants. And luckily in that genus, there's one species that's normal. This is a large queen. But the other species have ergatoid queens that are morphologically very, very different. They don't just have one inseminated queen. They actually have multiple queens that are laying eggs. And those queens that are not laying eggs start performing the role of minor workers. Because inside the nest, somebody's got to take care of the larvae. And these larger workers cannot. These larger workers, maybe because of selection pressure to capture larger centipedes, that's all they eat, have become larger. And now the queens form the inside the nest duties. So it's a very interesting story, basically launched because of DNA barcoding. Now for taxonomic provisions, of course, we include morphology. But at the same time, we can use DNA barcoding to actually delimit, actually, the unique barcode sequence for that species. And the more we do this in Madagascar, the more we're creating this DNA database in which you can start using simple sequencing to identify your species. That also means you can identify prey. There are some ants that eat larvae of other ants. We're beginning to understand what species they're eating by simply sequencing their larvae that they're capturing. And in the future, maybe ecologists who go to Madagascar can actually not even contact a taxonomist. They can simply sequence their ants and know what species they're working on. But is this helping us answer that one question? Can entomology and taxonomy be relevant in a country like Madagascar? We've been making great discoveries about ants as a whole. That's important for the scientific international community. But what about Madagascar? How can we compete with the birds? So to test that, I went to 4 sites in Madagascar that are relevant to conservation and tried to ask those kind of basic questions about the first questions you want to know about species diversity, species richness, and patterns of species turnover and endemism. So in those 4 collections, we took specimens down the traditional route. We mounted them, we labeled them, we databased them, we measured them, we identified them to species. This whole process rushed through, took about 6 months. We took a leg off, sequenced them, had all the data in 11 days. So we're comparing basically 11 days versus 6 months. We want to look at patterns of richness and patterns of species turnover. So we compared morphal species here in black with a very kind of crude measure of genetic diversity. And used kind of 3% cutoffs of genetic difference and 2%. There are much more sophisticated ways, but even these crude ways showed that you get the same ranking in terms of species and genetic richness, or morphal species and genetic richness. And in terms of ranking of species turnover, you get the same ranking. So in other words, in 11 days, we could actually tell the group that was organizing these inventories that which site held the most diversity and which two sites complement each other the most. And this is exactly what was of interest to the conservation group that was organizing this inventory. So there is a sense even that for that half the insect groups we have, where there's no living taxonomist, we could even sequence them and actually provide data. We could justify the large-scale collections if we were including these sequencing and would have even more data to evaluate these types of decisions. I'm not saying ants are special in that regard. They're special in the environment, but we want to include a broad spectrum if we want to create this Dow Jones Index environment. So we were able to kind of start creating this map of diversity through speeding up taxonomy, through this collections. Combine that with species modeling, we were able to create a map of diversity for the ants of Madagascar. And working with Claire Kremen at UC Berkeley, we were able to include ants in this large analysis of diversity patterns and come up with a strategy for preserving those remaining patches of forest. Colored in 3 colors here for levels of priority for conservation. And this is now the roadmap in Madagascar for conservation. And I'm glad to know that it does include ants at least. We should include more species. That does involve more effort by other taxonomies to help us. Now there's one caveat to this. Now I want to explain that if we would go to this world where we're just using sequence data to quickly give results to conservation groups while the taxonomists worked on their specimens, is that, let's go to Mauritius, where we tried to repeat the study. Now if you look at just all the species we collected, the place with the highest diversity is breeze. So you could say, well, that's the highest priority for conservation. But if we went through and weeded out all the known introduced ants, you get a different place. Actually, an unknown place that's not even in the reserve network in Mauritius. Forgotten place. And it actually is the last refuge for native ants. And that would not have come up if you didn't know which were the invasive ants. So to apply this kind of system worldwide, we'll have to find a way to actually create a database of the known invasives. But as it turns out, it's hard to identify invasive ants. In fact, it's locally a real difficult issue. So I began playing with some of the data to come up with a kind of tool for identifying introduced ants, non-native ants. Now we've noticed this pattern that over large geographic scale, introduced ants have low genetic diversity. So I'm attempting to try to come up with a measure that measures or compares geographic extent with genetic diversity. This is kind of a crude method, but this compares the mental R of genetic diversity and a pairwise of distance here. But to set up a gauge, we actually looked at urban habitats across Madagascar where we thought that we'd find the most invasive ants. And then we actually looked at known invasives and found that the known invasives fell in this category. So it comes out that basically, if your mantle R is less than 0.1, you potentially have an invasive ant. But what was interesting is that some of these points down here were actually endemic ants, or described as endemic ants to Madagascar or Mauritius. And we were surprised by that. Such as this one, Solenopsis mametu. There's actually a conservation program on this ant in Mauritius. I wrote colleagues saying, hey, I would hold off on this kind of promoting this Solenopsis mametu for now. They're like, no way. It's endemic, you know. Taxonomists said that in their paper. Well, I said, well, I'm suspect. We actually sequenced ants from Hawaii, which have only introduced ants. And there's an exact match in the sequence data from the ants of Hawaii and Solenopsis mametu. Now, what name applies to this? I have no idea. It's very difficult to put a name on this introduced ant. But at least now we start realizing that this is a really strong candidate for a non-native ant and should not be part of a conservation program in Madagascar. And right now we're trying to promote the development of this database of introduced ants throughout the world by sequencing ants from each potential invasives from each biogeographic region around the world. And this is what we need to actually then develop this kind of tool for just sequencing and applying that data, where we can quickly look at the sequence and pull out the introduced ants. And then the remaining ants would be native and can be applied for conservation. Well, I think I'm getting tired, so I think I'll stop there. And thank Phil for his, where it all began, the virus. And my team in Madagascar and all my collaborators on our current project, the ants of the Southwest Indian Ocean, as well. And Christian Peters in Paris for all the behavior work. And you, thank you. I guess we have questions now?
Yeah, we take questions before we leave. Yeah. Yeah, we have time for questions.
Howdy.
Hey, Truman.
So one of the things that I struggle with is you know, I could train up Kenyan PhD and master's students, and you know, get people, steal them from range science and get them interested in doing ecology and conservation. But then what? You know, where do they go then? You know, what's your thoughts on how you create or provide the employment opportunities for those people after you have lured them into being interested in taxonomy?
Well, I am the biggest hire of entomologists in Madagascar.
And you'll be dead soon.
I'm a taxonomist. I'll be dead soon. But we work with our PhD students, the ones that we're going through a PhD program, to make sure that they are marketable. Now, there's this amazing thing that created a boom for entomologists around the world. And that is the IFC and the World Bank basically follow a mandate called IP6, which requires them that if they want a loan of money, they have to actually do certain they have to follow certain environmental guidelines, which did not include insects at all until January 2012. And in preparation for this big change, we've noticed a lot of need by almost every mining company in Madagascar to hire entomologists. And this is where they're all going right now. They're leaving me in droves because they can pay. Now what they're required to do now is every private enterprise that receives money from the IFC, which they all do basically, they have to guarantee no introduction of any introduced species. And that's the first time they have to deal with ants. First time. So we're not being dealt with as environmental impact in terms of conservation. We're being involved. And we always say we have to do an inventory ahead of time. And then because we have to know what introduced ones are there, so we can monitor change in introduced species. So ports we're doing right now in Madagascar, pipelines, and mining sites and restoration. So I think in Madagascar, at least, what be the same in Kenya, that there is a market if you train them for that market. So that's the biggest potential market, besides going in to be a professor or work for the government in the protection system. Or because they speak English and can type and can write, they get a job in business, any business.
Going all the way back, I guess, to the early part of your presentation, when you mentioned the massive colony of ants you brought to San Francisco, you would feed them 25,000
Crickets.
crickets. Couldn't you feed them vermin meat of some sort? Throw on steaks or something? I don't know.
We thought about throwing in small children. There's a lot in the museum. But I don't think that would go over very well. So crickets are a great source of protein. In fact, you should all consider eating less meat and try to eat more insects. It's a sustainable, I think much more sustainable. In fact, it's the most efficient way to get protein into your system is to focus more on insects.
Was it cheaper to get that many crickets than to get nutrients?
Is it no? Well, actually, before I had that first order, I kind of warned the cricket farm, "Hey, I want you to know I'm going to order like a lot of crickets every day, and you can't miss a day because the colony collapse will happen." He said, "How many?" And I said, 25,000. And he said, "That's nothing." I'm like, "What? Who's buying crickets?" I go, "We have orders of millions and millions every day." I'm like, "Who's buying this?" Oh, we can't say. Yeah, who is it? I don't know. Is it like trout farmers or something? I don't know. But 25,000 is nothing. So they arrived every day in FedEx. What's that problem?
What's the etymology of ergozoid?
Oid is like a droid, like an android. And erg is
Yeah, work, sure.
Yeah.
Worker. Yeah.
it's like work like a droid-like worker. I wanted Google to come up with that for their new software. I mean, rather than Android, I thought ergoid would be better, but they didn't choose it.
Yeah.
Gandromorph is a genandromorph, right? Jim?
So if you were head of NSF research funding, I mean, what's a guiding principle for how much you put into taxonomy and field biology versus molecular and so forth? I mean, everybody, is going after whatever they can, get. But in fact, there's some, sort of guiding principles there on how you maintain some sort of taxonomic presence and so forth versus the molecular and so forth. I mean, what's your views on if you were head of NSF, how would you decide how to appropriate different levels of funding?
Well, this is what I would do if I was in charge of NSF. Now, if you go down to any of the largest labs right now that are doing molecular work, and like at Mission Bay, at UCF, University of California at San Francisco, and you go and talk to the graduate students. You go, "What are you going to do when you finish here?" "Oh, I'm going to do a startup." "Oh, I'm already got a startup lined up. I'm going to start my own company." So it's like molecular work has already been so far advanced that they actually have created cottage industries already that are sucking up all the students we're producing. So that seems like they're not hurting at all. If you go in that area, maybe more so at UC Davis, but not down there. Now, if you go into a taxonomy lab, you'll see that they're really hurting. But we have the same potential to be creating cottage industry of growth and business around taxonomic work if there was proper investment. And that investment would be in actually understanding. The problem is that we're at such the bottom of the curve of understanding the environment. Everything we do impacts the environment. Everything could be done in a way that even big mining companies, if given the choice, given the data, they would make decisions that may reduce their footprint or their impact but they have no way of getting data now. There's no industry out there that's being driven by the knowledge base of understanding the world because we're so far behind. So I think there's potential of investing in taxonomy, like $1 billion instead of a telescope. And that would launch a change in taxonomy in the sense that there is cottage industry actually then also equally supporting those endeavors.
So how do they make money? How does this cottage industry make money?
Because we'd start beginning to, at various levels, understanding the environment. And therefore, just simply environmental impacts would involve holistic approach. Now we can almost imagine this, right? We could take a malaise trap in Madagascar, pour off the alcohol, sequence the water that's in there, the alcohol in there, and get a list of ants that were in that malaise trap because we have this database. Now imagine if we had this database in the United States, for every forest, for everything that's living in the United States. That means we could even have like a machine out in the forest that would sample once a month or once a week, and it would like send a message, oh, we just got intercepted an introduced species, or we're just giving a printout of species every week. Understanding would be so critical of our society in the future that this will have to be part of every business that impacts the environment, every road, everything. And that will start driving the need for sampling, and that would create, in a sense, continuation of the sampling regimes that we need for the world around us. That's like a futuristic sense, but it may not be that futuristic if we start sooner. Taxonomy will also be important for actually all the stuff we're going to find in there. And you'll care about, if you could actually find information, as soon as you're stung by something, you want to know what it is. And that's impossible almost right now. The average public, if they're stung by something, cannot find out about what it is. But in the future, they should be able to.
I just wanted to reiterate your last point because I think it's really, really important because I don't think we have a very clear vision even as biologists. I think we try to have a clear vision, but it's hard to cling to it because we're not from a culture or species that any longer has the clear vision we might have had 20,000 years ago, and we did have 200 years ago when we had much more intimate relationships with other living things. So the need for taxonomy, a lot of what you're talking about, and a lot of what is, I think, perceived clearly is, well, we don't even know the names of the things that are going extinct. So a lot of the growth need that you're referring to, understandably, is for conservation of biodiversity and protection of endangered and threatened taxa. But beyond that, we often say we need to conserve these areas, these biomes, these natural communities, because we can get important things out of them, like cures for cancer, et cetera, et cetera. What we don't really, in general, we know very little about what any member of any species does most of its time, spends most of its time doing, or what its important interactions are. We don't have any sense of, we have very little sense of the behavioral ecology or the important events on a day-to-day basis for most things that we study. We just don't have the capacity or the sensibility and to grow in that direction so we can enrich our relationships with other taxa. Obviously, it will require a lot more data and richer sensibilities, but it's that, I think growing in that direction is fundamentally based upon the sort of thing you're talking about. Not just beyond just naming things. Naming is the first step, and then getting to know them better, and then having a biodiversity not just of taxa, species, genes, but also biotic associations, or particular kinds of behaviors, or particular kinds of relationships with our own species. That all fundamentally could grow from the sort of vision that you're promoting with what you're saying. That leads to many cottage industries.
Can you think of maybe like one sort of concrete example where this type of system could be attached to an industry, say here in this country, where you could show that people could make money using this, or have it be associated with somebody making money? Maybe they're not directly making money out of it, but for some regulatory reason. Something really concrete so that it could sort of almost be like a demonstration project, say, so that people could who don't even know what taxonomy is could just think, oh, well, if this worked here, maybe it would work here.
So every company that has an impact on the environment is required to actually monitor something. So they're monitoring their stream quality. If they're a clothing factory, they have big impacts on streams. They all, like Gap, I've met with Gap to talk about this actually in Madagascar, because they have one of their companies in Madagascar, one of their factories rather. Anybody who has a need to monitor, anyone that has a need to evaluate their impact on the environment through study can benefit from this. And it's interesting, the US is not doing this except in stream monitoring, I don't think. But actually, the number one contributor to ants of Africa is actually an environmental impact consulting firm who's actually incorporated barcoding because he finds he gets his bids better because he can offer barcoded data. So now, and he just does ants. So he runs around Africa doing environmental impacts on ants from Tanzania to Kenya to Angola and in South Africa. And we get all this barcode data, which is great, and he contributes his specimens, but his bottom line is barcoding is important for his project because it helps him deliver data to his companies. So he's now has to voucher specimens better and database them, all this stuff that maybe an environmental consultant firm won't be doing. But to barcode them, you have to participate. That means submitting the data for sequencing. So he's now the number one producer of ant data for Africa, and he's a consultant for an environmental impact company. So that could happen more and more. I see it happen. Interesting, it was Africa that did it first.
Okay, well, let's give Brian another.
And Uganda, ant course, next year.
And to commemorate your time here, we'd like to give you this, this plaque. And thank you again for a great talk. Thanks. Well, it's time to eat, so let's go.